I'm very excited. I have a critic which I'm going to answer today with this short program on the iron preservation theory. First of all, he claims that I have no independent supporters. He says, I cannot take what Armitage says as fact because he has no independent supporters. Well, surprise, surprise. This is a hot potato in the biological community. This is not something that people want to pursue because it's a career ender. It's a career killer if you go after soft tissue and dinosaur remains. And so my own case is a case in point. The fact that I was running $1 million laboratory at California State University, Northridge, a thriving, booming laboratory if you listen to some of the professors and read their writings from that biology department. And yet 2 weeks after my paper appeared online, I was terminated, given my walking papers out of there. But it's no surprise that I have no independent supporters. In fact, Mary Schweitzer, at the beginning of her work, had no independent supporters. Surprise, surprise. In fact, if you look at the article entitled Mary Schweitzer's Dangerous Idea, I think it was in Discover magazine, she recounts the story where one of the reviewers of her paper wanted to have nothing to do with her paper, her report, and so she asked the reviewer, well, what evidence would you accept? And he said, well, none. This is from the 2004 American Society for Nutritional Sciences Journal of Nutrition, written by Dr. Joe McCord, and the title is Iron, Free Radicals, and Oxidative Injury. And he's talking about cell injury. And here's a quote from the paper. Free iron is a loose cannon chemically. One of the most devastating actions of free redox active iron within the cell is the initiation of lipid peroxidation. It leaves in its wake dysfunctional membranes and cell death. And so there you have a researcher in the field who's studying these lipid membranes in cells and who states clearly that free iron in the body leaves in its wake a trail of destruction and death. So for him to claim that I ignore that iron generates free radicals is really an abject ignorance of what I have said and what's in the literature. Now, he goes on to talk about the fixation process. You're talking about iron nanoparticles. Now we're talking things that are nanometer sized. In fact, some of these are measured out about 100 nanometers in size, these iron nanoparticles, and according to Schweitzer, they perform the fixation. They cross-link the proteins. Now, again, I mentioned that the molecules that we use in fixatives to fix these proteins are angstrom sized. And the reason they are able to fix these proteins is because they can surround the entire protein because they're angstrom sized, and they can fix the tissue by that cross-linking process. And so you're expecting a nanometer sized, in fact 100 nanometer sized iron particle to do the same fixation that an angstrom sized molecule does. It's kind of like using a trash truck to staple your papers together. And so nanometer size, 100 nanometer size iron particles don't do the trick. Now let me make another point about this supposedly fabulous system of iron preservation of tissues. If iron preserved the tissues for 70 million years with the kind of stunning preservation that I'm photographing from these dinosaur soft cells, if iron could perform that, then why isn't it in my protocol manual? Okay? I'm a member of the Microscopy Society of America, have been for many years. I'm on the board of directors for the Southern California Society for Microscopy. Nobody that comes to present at our meetings has ever talked about iron preservation of tissues. And so if it is, as you say, a fabulously efficient fixation protocol that can fix proteins and tissues for 70 million years, then they should be in our protocol manuals. In fact, I should be able to find it in the latest edition of Weider's Functional Histology, which is the textbook for studying histology and tissue relationships. And of course, this goes into great detail to talk about fixation. So why isn't iron preservation in this textbook? Why isn't it in any of the textbooks or protocol manuals that we use for microscopy? Number one. Number two, why aren't the cryopreservation companies using it? You could run down and find a patent attorney, my friend, my critic friend. Run and find a patent attorney and get yourself a patent pronto for iron preservation of tissues, because the cryopreservation banks would buy that from you and you'd be a multi-billionaire. When you look at Schweitzer's explanation, her theory that iron preserved the tissues, and you study the materials and methods section, which is a little difficult to find. Now, none of those conditions, it can be said, are indicative or related to what happened at the Hell Creek. When those things were buried at Hell Creek, there were no ultracentrifuges, there was no anticoagulant available, there were no molecular filters, and so just to get to the point where she could begin the experiment, she had to employ all that laboratory technology, and then of course she left it on a laboratory bench at room temperature for 2 years in the absence of water, which is abundant at Hell Creek, the freeze-thaw cycle, the heating of the Montana summer, and all of the microbes and scavengers and insects and everything that was fully involved in those specimens, and particularly my specimen, the Triceratops horn, fully open to the environment, highly vascular, and so she had to remove all that from her experiment in order for her experiment to work.